All right. Pleasant good evening to everyone. Um, this is the first day of our 6-day faculty development program. So by this time, we are starting our program with the inaugural session. Let me start with the welcome address of the session.
Again, once again, over here and stick it over here.
It's my honor and privilege to extend my warm welcome to all of you who have joined this faculty development program. As the coordinator of this program, I'm really delighted to see such an enthusiastic gathering of educators, scholars, professionals who are committed to continuous learning and academic excellence. And first and foremost thing, I like to extend my sincere gratitude to our college management, principal, dean, and our head of the department for their unwavering support and guidance in organizing this event. A very special thanks to the resource persons of their day, Dr. Ronaldo Anuf and Dr. Batmanabhan, who have taken their time to share their knowledge and expertise with us. Their insights will definitely enrich our understanding and inspire us in implementing innovative teaching and research practices. I also extend a warm welcome to all the participants. Your enthusiasm and willingness to engage in this program reflect your dedication to the professional growth. I'm confident that this FTP will serve a platform for knowledge exchange, collaboration, and the development of new perspectives in our field. This program is designed to equip us with the latest pedagogy strategies, research methodologies, and technological advancements that can enhance our teaching effectiveness. I encourage each one of you to actively participate and ask questions, make most of the learning, most of the sessions very interactive, and have a good learning experience. I once again welcome you all and I wish you a very grateful and fruitful, engaging faculty development program. And I will also expect your kind cooperation for the success of this program. And thank you one and all. And followed this welcome address, we'll have felicitation by our head of the department, and then an overview of the sessions by Dr. Ian Prabhu. And at the end of the session, we'll have an introduction to the session 1 resource person. Now, I welcome my our department head of the department, Dr. N. Nageshwari, to felicitate this gathering. Ma'am.
Thank you, sir. Am I audible, sir?
Yes, ma'am. Yes, ma'am.
Thank you. A pleasant good evening to one and all. Respected dignitaries, esteemed resource persons, distinguished participants, and my dear colleagues, a very warm and heartfelt welcome to each of you. It is truly a great privilege for me to address this gathering on this occasion of ATAL Faculty Development Program on revolutionizing healthcare and biotechnology, the role of AI and computational techniques on behalf of Sedu Institute of Technology and Department of Biotechnology. As we step into this FDP, let us reflect on a profound thought by Thiruvalluvar. Katrathu kaiman alavu, kalladadu ulagalavu. What one has learned is only a handful of sand, what is yet to be learned is as vast as the world. So this timeless wisdom perfectly resonates with the theme of our FDP. As you all know, nowadays, artificial intelligence is no longer just an emerging technology. It is entirely a game changer. So it is making various incredible innovations in the field of science, in the field of environment, in the field of healthcare, in all the domains, it is playing a major role. So while AI is making incredible strides, its true potential can only harnessed when the domain expertise like biotechnologists or computer science engineers or any other biomedical engineers or any domain expertise meets this computational power, then only the gaps can be filled. That is the synergy between the artificial intelligence and the biotechnology is already leading to remarkable advancements in the field of healthcare, like AI-powered cancer detection tools and smart microfluidic devices and wearable devices with the help of AI. Like that, various technologies has been revolutionizing the world of biotechnology as well as with the help of the artificial intelligence. So this FDP is going to be a wonderful opportunity for all of us. I extend my deepest appreciation and gratitude to AECTE Training and Learning Academy for organizing this highly relevant and futuristic program, and also my sincere gratitude to the exemplary resource persons for all these 6 days for sharing their expertise, and to all the participants for their enthusiasm and commitment to learning. As we join together for getting technological transformation, and let us remember that it should be an initiative for collaboration and to share our curiosity, and there should be a perseverance for continuing our innovations. So definitely this FDP ignite new ideas and inspire research collaborations, and ultimately contribute to revolutionize the healthcare and biotechnology field for getting a better tomorrow. Once again, I extend my heartfelt welcome to all of you and best wishes for a productive and enlightening FDP. Thank you all. Over to Dr. N. Prabhu, sir.
Good evening, one and all present here. Once again, we are welcome you all for the 6 days ACT ATAL FDP program. So about the program, we are planning 6 days, 13 sessions. The session 1 start today. The title is Decoding Genetic Variants, SNP Analysis Tools and Techniques for Precision Medicine by Dr. A. Ronald Anup. Second session is sustainable technologies in antibiotics degradation, path to a safe environment, handled by Dr. V.C. Badmanabhan. And third session is tomorrow. The topic is biomarkers and personalized drug development by Mr. K. Chandru from Bioanalytical Department, Sinjin International, Bangalore. The fourth session tomorrow, the topic is Role of Functional Foods and Nutraceutical in Human Health by Dr. S. Babuskin from D.Y. Patel International University, Pune. The day 3, session 5 is Impact of Machine Learning in Clinical Data Analysis, Mr. S. Jenny. And sixth session is Key Aspects of Scaffold Design in Tissue Engineering by Dr. S. Uday Kumar from Department of Chemical Engineering, IIT Tirupati. Session 7, the fourth day, Topic is repurposing cholesterol lowering drugs in the treatment and management of monkeypox from Dr. R.S. Suresh Kumar from Arab Means University, Southern Ethiopia. Session 8, Role of Computational Tools in Optimizing the Production of Biodiesel from Microalgae Biomass by Dr. S. Renganathan, Professor, Department of Biotechnology, Anna University, Chennai. Day 5 is a role of computational and programming in clinical data analysis by Mr. G. Viji from Pune. And say session 10 is CRISPR-Cas system for combating AMR by Dr. INR from US. Day 6 topic is the role of big data in personalized medicine by Dr. A. Rodand Anuf. Session 12, the role of AI in cancer diagnosis and treatment by Dr. Mr. S. Jenny. Last session is development of diagnostic against infection and antimicrobial resistance by Dr. V.C. Batmanoban. So this is a plan for our 6 days ATAL FDP. In the FTP, we have a feedback session. We have everyday feedback sessions there, and as well as finally, you will going to complete the assessment test in the ATAL portal. So finally, you can get the certificate successfully, and we are expecting more coordination and interest from your side. So thank you for the valuable time. So thank you.
Thank you, sir. Before winding the inauguration session, I would like to call the resource person, head of the department, and co-coordinator, Dr. N. Prabhu, to switch on your video. Let me have a snap. Ravi, sir, you snap. Right. Thank you. So we are at the end of inaugural session. We are going to start our first session by Dr. Ronald Anus. Let me welcome Mrs. Vanaja to give a brief introduction about our resource person today.
Hello.
Hello. Good evening, one and all. I thank God and universe for giving me this opportunity to introduce our eminent speaker.
Vanaja, kindly unmute yourself and
Hello, sir. Am I audible?
yes, ma'am. 1 second, ma'am. I request all participants, please do not unmute yourself while the session is going on. Thank you. Yes, ma'am, please proceed.
I thank God and organizers and the universe for giving this opportunity in introducing our eminent speaker for the session 1 Dr. A. Ronaldo Anup. Dr. A. Ronaldo Anup is a Director of Research and Development at Silicon Labs Private Limited, based in Marthandam, Kanyakumari District, Tamil Nadu. He holds a PhD in biotechnology from Anna University, Chennai, with a focus on developing optimal strategies for bioethanol production. Dr. Anup completed his Master of Technology in Biotechnology from University Departments of Anna University, Tiruchirappalli. And Dr. Anup has expertise in biopolymers, bioactivity studies of phytochemicals, and drug discovery using bioinformatics tools. His research aims to uncover innovative solutions for cancer, inflammatory diseases, and Alzheimer's diseases also. And he made a significant contribution to various international journals, publishing over 23 papers and garnering an impressive cumulative impact factor of 72.44 and over 1,200 citations. Dr. Anup's work on microwave-assisted extraction and molecular docking studies has made a notable strides in biofuel production and medicinal chemistry. Before his current role, Dr. Anup worked as a research associate in Shaili Biotech Private Limited, where he optimized media for bioethanol production. He then served as an assistant professor at Kamaraj College of Engineering and Technology, Virudunagar, where he dedicated 10 years to advancing the biotechnology department. His academic journey also included a stint as a lecturer at Bharidasan University, Tiruchirappalli. Dr. Anup has filed several patents, one of which was granted for innovative method for biological lignifcation of Prosperous jurifera. In addition to his research, he has edited a book on environmental sustainability and human welfare, and also authored chapters in renowned international publication. Dr. Anup has led and contributed to several funded projects and national level conferences, further establishing his leadership in field. His academic and industrial experience have equipped him with a deep understanding of biotechnology applications, but particularly in bioenergy, biopharmaceuticals, and computational drug discovery. With a strong commitment to research and development, Dr. Anup continues to shape the future of biotechnological advancements in Silicon Labs, making valuable contributions to both academia and industry. I heartily welcome you, sir.
Thank you, ma'am. Thanks for the introduction.
Yes, Professor. Your audio is clear.
Yes.
Now the forum is yours. You may please enrich the audience.
Thank you, Professor.
And for your kind information, our participants are diversified all through the India. We have participants from North India, South India as well. We have around 34 universities and colleges are participating in this faculty development program, and a mixture of population with a mixture of departments are there. We have participants from biotechnology, computer science, electrical and communication field, as well as biomedical engineering, and few participants from information technology, chemical engineering, and all other related departments are also participating in this program. So your input will enrich all diversified platform here. So now the forum is yours. Thank you, Professor.
Thank you, Doctor. I'll share my screen. Please help me whether it is visible.
Yes, sir. Yes, sir. Your screen is absolutely fine. That is highly visible. Yeah, presentation mode is all. Yeah.
Thank you, Professor, and thanks for the team. Thanks to the faculty members from Sedu Institute of Technology for giving me an opportunity to interact with this diverse group of participants. And today I will be presenting an insights on SNP analysis tools and techniques which are currently used for precision medicine. I hope my audio is clear. Yes. Dr. Amal?
Yes, Professor. Your audio is very clear. Very clear.
Thank you. So, in today's context, we know precision medicine is ruling the country. Our nation is taking wide steps in establishing AI-related advancements, especially in the area of precision medicine and personalized medicine domain. So to highlight a few important upcomings which have come up in last 3 to 4 months, a very important advancement is related to the precision medicine concept is that Siemens Healthineers, we all know it is based upon the Siemens group. They are currently collaborating with IISc, and they have started a new AI lab which is completely dedicated for precision medicine. It is a part of their academia institute interaction. They have dedicatedly opened up a new lab which is completely focused on precision medicine, especially for cancer. And we can also come across a recent news which was published in Outlook in the end of 2024 that states that AI and precision medicine is going to be the key players in non-communicable disease management. So for most of the non-communicable diseases, it is no longer going to be one drug which suits best for all the patients. It has to be all the market leaders in the pharma segment. They are now moving towards this precision medicine, and they are trying to bring up some novelty, especially in all the non-communicable diseases. And there is one more important advancement which are published very recently in Hindu, especially in the oncologist conference which held very recently. Most of the oncologists now believe that it is precision medicine which is going to be the right solution for the patients who are being subjected with cancer. So these are some of the news which we are coming across in recent days related to the advancements of the precision medicine and dependence of precision medicine by both the people from the research, from the industry domain, and people from the medical domain. I'll just share you an insight of how this industry is going. This industry is going in a remarkable rate at a CAGR, current annual growth rate of 8.2% over the last, and the projected growth rate is approximately 8.2%, where personalized medicine diagnostics, personalized medicine therapeutics, personalized medicinal care, and personalized nutrition and wellness are going to contribute to this growth in approximate size of around $529.3 billion in another 6 years of time. And I've also given you an insight of the market share of major countries across the globe who are contributing to this exceptional development in the field of personalized medicines and markets. I'm also happy to share you a data. I was actually thinking to give you a clear glimpse about the data which has been published very recently. But okay, I got a data which was published in the early 2021. Key players, starting from 2021, all the key players in this field have focused on development or focused on both research and identification of suitable areas, especially for bringing up precision and personalized medicine in the sector of pharmaceuticals. Starting from Novartis, Pfizer, Catalent, Johnson, Roche, AstraZeneca, all the companies who are the pioneers in the pharmaceutical sector have started to dedicate a sector completely dedicated for personalized medicine. So this itself shows the unique potential of personalized medicine and how it is going to rule our industry in the next couple of years. This is not the scenario, right? Whatever I have told you is maybe the global scenario of how things are changing over the globe in the previous era. Now I'll just let you know how it is moving on in India. There are so many startups who are coming up with different innovative ideas. To name a few, we know Xcode Life Sciences. They are coming up to provide solutions related to all lifestyle diseases like coronary atherosclerosis, diabetes, obesity. They are trying to build up a saliva kit which can easily determine the allelic differences between the individuals and prescribing a personalized medicinal approach for them. The second important company which is gaining attention recent days is Nutragene. So Nutragene is our country's first commercial genetic test which is completely dedicated for type 2 diabetes. So they are currently doing a type 2 diabetic genetic scan, especially a DNA testing services, which is widely to access all the risk factors for type 2 diabetes. It's just a swab-based tester. And we also have a major company, Avasthajan, which was also a new company which has which have been focusing on several milestone projects in India. They are also come up with a system biology based study for, especially with, they have done their major study in a part of the population on identifying the genetic basis of increasing the longevity and age related disorders. And they, with those data of genetic diversity, they have also come up with a model to develop, means to identify biomarkers in patients for identifying the age-related disorders. And they have also come up with some novel solutions personalized for people to improve their longevity of life and to overcome the risk of age-related disorders in them. So they have enrolled, spent around 125 crores in this project starting in 2007. And now they are in a very established position, demonstrating their leadership in this particular domain. Right? And there are several issues and several initiatives taken from the government of India. Like in 2019, we all know that Dr. Harshvardhan, the then Union Minister for Science and Technology and family, Human and Family Welfare, started with a unique portal of Nidang. Right? They tried to identify different national level inherited diseases administration Kendra, where they tried to map up the human genetic diversity in 20,000 sites across India. And now they have come up with a wonderful data. I'll show you in the later slides. And we all know that there is Ayushman Bharat, the digital mission coming up in India, where under the leadership of our Prime Minister, the efforts have been taken to come up with some digitalizing the human medical data, which will actually help us in taking things further. And we all know Accenture, a very interesting IT company, IT-based company providing IT-based services. They are also into it. They have also entered into the precision medicine market. They are trying to come up with some novel solutions for the public, especially with related to the genetic characteristics data across different therapeutics domains. And things are not stopping there. And we all know that we have a Viksit Bharat plan of 2047. Our country is preparing for the Viksit Bharat plan 2047, and they have established precision medicine and use of artificial intelligence in diagnosis and treatment as the major area which is going to revolutionize the healthcare sector. And there are contributing partners from different institutes, starting from Institute of Life Science and Technology Information. They are taking the key role, leading the emphasis on point of care diagnostics and treatment, especially with the genetic diversity data, which is being taken up in different parts of the country. And they are preparing the action plan, and their efforts have started to yield good results. We all have witnessed that our Genome India data has been published last January 9th, 2025, almost 1 month before. It was launched by Dr. Jitendra Singh, the Honorable Minister for Science and Technology. He has released the Genomic Data Conclave, and I think you are all aware that DBT has also come up with a plan of, has started to release a translational research fund, and the call is open. So anybody who is interested, any researchers or academic institutions or industrial partners who are interested in working with this genome data of India, which is available right now, they are able to provide funds for that. India is coming up, DBT has requested for proposals for people to work with this genomic data and their translational research scheme. The call is open, so any of the participants who are currently interested, any faculty members who are interested in applying for this, or also can also make use of that opportunity, because all the researchers and industrialists out there are now going to utilize this data in development of personalized medical alternatives, personalized medical solutions to patients for different conditions. So you can take use of this opportunity to establish your field, to come up with some novel data, come up with some novel findings with the genome data, whatever is available. More than 20,000 sites in India is dedicated for this genomic data collection. It has been compiled perfectly for the last 4 to 6 years, I think. It started in 2019, Nidan Kendra, and it was published in the year 2025. Now this data is completely available. It is free to access, thanks to the efforts taken by the Department of Biotechnology and Ministry of Science and Technology. And this data is now available for all the industrialists out there to come up with some novel solutions to address the Indian population diversification and the steps which need to be taken by the researchers to provide a personalized solution for almost all the non-communicable diseases for which the data has been collected. So this data is going to give us a new dimension, a new paradigm shift in the treatment opportunities which are currently available in our country. And our world, means India, is going to witness a huge change, or a huge translational change in the way we treat diseases. So this is the recent advancement. I also would like to thank the organizers for selecting this topic for the presentation in today's conference. This is the recent buzzword what the biotechnologists out in India who are especially doing their dedicated research in bioinformatics were looking for, and they are working tirelessly for coming up with some novel outputs from the data which is currently available in the genomic database. Okay, so what is about today's presentation? Which area am I going to concentrate on today's presentation? So today I'm going to completely cover up on the genetic diversity and how it is influencing on the pharmaceutical sector, how it is holding a grip over the drugs what we take, and what information is further required to provide a personalized treatment option for all the participants. We all know that human genetic diversity is at a larger extent. You can see that there are several countries where the genetic variability is extremely high, and we are in a safer position. We are in a mid-range, and compared to the global genetic diversity. And there are several reasons behind this genetic human variation and how this has happened. And I'm not going to enter into a deeper part in this area because that is not the context of our topic. But I'm going to touch on this area because this is the major area which needs immediate attention and which is requiring the attention of all the scientists out there, especially the multidisciplinary team which is joined today, to have a look at this data, how to use this data, and how to come up with some novel solutions for patients who are subjected to different diseases. We all know that we are subjected to genetic diversity. There are several reasons behind that. It could be either a sequence variation or a structural variation. And today, in my context, I'm going to highlight on a very interesting area that we call it a single nucleotide polymorphism, which is a sequence variation based changes in your genetic segment, which is creating an impact on the drugs what we take and our treatment regimen, because no two persons are going to be the same. So this genetic diversity in our population can bring huge differences in the pharmacokinetics of any drug what we take. There is going to be a variation in the effect of the drug or the influence of the drug in different patients, and this single nucleotide polymorphism, especially in the areas like in your drug metabolizing enzymes. Of course, we'll have a few examples in the upcoming slides. SNPs in your drug transporters, SNPs in your drug receptors, SNPs in your ion channels. If there is any SNP or single nucleotide polymorphism in these areas, either the drug metabolizing enzymes or drug transport, drug receptors or ion channels, it is definitely going to impact the society. When there is a modification in these areas, of course, it is definitely going to create the way in which our body is metabolizing the drugs. And of course, that will lead to different effects of the same drug on different population, right? Which is the current scenario. So before that, for us, Dr. Amal highlighted, since there is participants from diverse background, I just want to tell what this genetic means SNP is all about. SNP is a genetic mutation, right? A single base pair will be changed. We all know that the genetic information is stored in our DNA, right? So there are several reasons behind mutation of our gene sequence. So imagine that there is a single nucleotide polymorphism which has taken place here. That has led to a change in the nucleotide position here. Instead of a CG, we are getting an AT base pair out here. This small single change which is happening in the DNA sequence is definitely going to create a change in the protein makeup of the patient. And when the protein makeup of the patient changes, automatically the influence of the drug on the target protein is also going to change. So this small change, this single nucleotide change in a patient setup is going to create several damages, right? Okay, so talking about this SNP, this is not that all the SNPs are dangerous. There are different types of SNPs, starting from non-synonymous SNPs, synonymous SNPs, nonsense SNPs, and intronic SNPs. Of course, intronic SNP is not going to create any impact, so it is not going to create create an impact, but it is occurring in the non-coding regions. We have nonsense SNPs. They just go and create a stop codon. So what happens is that protein will not be generated, that it will try to produce an incomplete functional protein. So naturally that protein will be destructed, or it will be subjected to digestion, and it will be sent out, or in some cases it will not yield the final result. Non-synonymous SNPs, they are the target these days. This is the huge problem these days. Synonymous SNP, there will be an SNP, but there is no change. There is a change in the DNA sequence, but there is no change in the amino acid sequence. Since there is no change in the amino acid, I tell you why. Since there is no change in the amino acid sequence, it is not going to create an impact on the structure of the protein, and definitely it is not going to create an impact on the drug and target relationship. But something which is going to create a greater damage is the non-synonymous SNP. When the DNA changes, the codon changes. When the codon changes, automatically the amino acid which is going to code for that particular protein is going to change. And when the amino acid changes, automatically the structure and the function of the protein is going to change. So in today's context, not only in my presentation, but the nation again is going to focus on this area, identifying the non-synonymous SNPs associated with the different diseases, and especially that so far we have been, we are also been working in this particular area for last couple of years, but we were actually utilizing the genomic data which was published around the world. But we were not given access to the data because we didn't have a data of our Indian population. But now Indian population data is available, and definitely we are very much interested in working up with this data, especially with the non-synonymous SNP data which are currently available there, because that is a real genetic variant which is creating a huge damage to the population, especially in terms of drug-protein interactions. I told you that there is a concept of non-synonymous SNP, right? So when there is a mutation, I told you that when a single nucleotide is changing, right, there are two options, right? Valine, we all know that it is coded by GTC, right? So instead of GTC, if it is going to GTG, again it codes for valine, right? Instead of C, if the position is changing to G, it codes for the same amino acid. This amino acid we call it as valine, because we all know the genetic codon, thanks to Hargobind Khurana for cracking this genetic code concept. And in the case of valine, right, we know we have 4 different combinations, 4 different codon concepts which codes for the same. So any single nucleotide change which is going to code for these, means if it is going to yield up any one of the positions, say in the third position, if it is going to be U or C or A or G, it is going to be valine again. So if any changes in the nucleotide position yield the same amino acid, right, the amino acid stays the same. The amino acid is going to stay the same, then it is not an issue. It is called a synonymous change or a silent change, because only the DNA, there is a change is witnessed at the DNA level, but at the codon level or at the amino acid level, you are not going to face any change, because still it is going to be coded by the same amino acid. So the function of the protein is not going to change, and the structure of the protein is also not going to change. So the drug target interaction is not going to influence. But problem comes where instead of T, if you are expecting a C there, then automatically in place of valine, we are going to have alanine out there. So if it is going to be alanine, automatically the amino acid has changed, and the amino acid is going to change. This condition, we call it as non-synonymous change. We are going to yield an amino acid which is completely different, which is not going to yield the same result as that of the previous amino acid. So when this amino acid change happens, when such a non-synonymous amino acid change happens, automatically this will lead to change in the structure of the protein. When the structure of the protein changes, automatically it will lead to a different type of variant response for the drugs which you are going to take. And this will have a definite impact on the pharmacogenomic drug profiles. It will have a definite impact on the genomic variant, especially in terms of drug efficacy and drug toxicity. And it will be also have a definite impact on the patients who are subjected to these forms of therapy. So an example will actually help the participants to get a good idea over that. We all know that there is a drug called as clopidogrel. Right? Clopidogrel is normally preferred for patients to prevent unwanted blood clots when the patient has come up with a severe heart attack. Doctors will normally prefer to give clopidogrel. So clopidogrel is an interesting content to take. So they normally prefer clopidogrel. So what happens with clopidogrel is that when you are subjecting the patients for clopidogrel, the blood clotting will be stopped. So when blood clotting is stopped, automatically the pressure of their which is built inside the heart will be reduced, and it is a safe kind of treatment which can be provided to any patient who has been subjected to coronary artery disease or acute coronary artery disease. So this is with respect to the genetic polymorphism which has happened in patients who are subjected to clopidogrel. We all know that there is a particular enzyme inside our system we call the cytochrome P450 oxidoreductase. Shortly, it is called as CYP. I'll also give you a glimpse of what the CYP is all about. And there are a group of population which people have witnessed that there is an isoform variation, especially in CYP2C19, right? So whenever a patient, due to several reasons, have got a CYP2C19 loss of function, they are not susceptible to clopidogrel. Clopidogrel is not going to be the suitable medicine option for patients who have undergone a mutation, especially a non-synonymous mutation in CYP2C19. So whoever is having a loss of function, they are not prescribed with clopidogrel. Instead, they are being prescribed with the ticagrelor or prasurogel. Of course, most of the heart patients know about the significance of the drug, and it is creating a several impact on the patient, right? So many drugs, especially if for abroad, FDA, as per FDA guidelines, in clopidogrel, it is prescribed to indicate that this drug is not suitable for CYP2C19 loss of function, right? In particular, genetic mutants in CYP2C19, clopidogrel is not suitable. So what medical company means hospitals are doing is that before prescribing clopidogrel, the patient has to be subjected to genetic testing and made sure that they don't have the CYP2C19 loss of function. And only after ensuring that the patient is not subjected to CYP2C19 loss of function, the patient is being provided with clopidogrel. This is a good example because it is a deadly disease and the patient needs high time care. No compromise has to be given on the drugs or whatever the solutions what the patient is subjected to take. So that is one thing. The second important area which we are concerned, this is not only the case with clopidogrel, but there are several drugs here scientific community which is actually not working on such population. Drugs for MOA, Alzheimer's, drugs for epilepsy, drugs like proton pump inhibitors, estrogen blockers, beta blockers. There is a list of drugs which does not suit well when your CYP2C19 is being attacked by mutation, especially single nucleotide polymorphism. Where there is a chance automatically this enzyme level is going to go down. When this enzyme level is going to go down, it is not creating the same impact on the patients, right? Okay, another good example which I can give you people is a very important drug which is being prescribed for blood pressure. That is metalor. Means most of you are also would have come across this. This is a drug which actually twisted the scenario of personalized drug medicine. So this is the first study which has been reported on pharmacogenetics and genomics that cytochrome P450 2D6 poor metabolizers will have an adverse effect of this particular drug. This particular enzyme, cytochrome P450 2D6 isoform, is about to metabolize this protein. So with an assumption that this drug will be metabolized, it is being prescribed at a higher concentration to the patients. What happens is that whenever a patient is subjected to cytochrome P450 mutation, definitely the concentration means his enzyme is not going to be functional. When his enzyme is not going to be functional, the drug is not metabolized. Instead, the entire concentration of the drug which he is going to take is now going to his target site. When the concentration of the drug increases in the blood, it is yielding 3 to 10-fold higher concentration inside your body, which is leading to several side effects, which has been reported in a very thorough state. So whoever new to this field, if you are finding difficult to understand what CYP enzyme is, CYP enzyme is a good enzyme which is present in your liver, right? So the function of any drug what you take, it enters inside your gastrointestinal tract and it gets absorbed, right, to your intestinal cell wall layer, and it reaches the plasma protein. The plasma protein will initially take it to your liver. So this is the site where your CYP enzymes are present. And their role is that any foreign drug enters inside, any foreign chemical enters inside, if it is witnessing it, it has to send out from your system by metabolizing it. So with that in mind, usually doctors will be prescribing a higher dosage, right? Even though if your drug is metabolized, at least some part of the drug is reaching the target site. Problem is clopidogrel is that it is not metabolized, so it is not reaching the site. Problem with metoprolol is that it is provided at a higher concentration. Again, it is not metabolized, but it is reaching the site at a very higher concentration. That is leading to several impacts on the patients, right? So these are the different adverse effects which are being caused in poor metabolizers, starting from asystolic problem, tachycardia, hypotension, cardiac decompression. Several adverse side effects have been witnessed in the patients who are poor metabolizers, and this was brought to the notice, and this drug is also now being stopped from prescribing for the patients, especially when the patients are prescribed this particular drug, they are being genetically tested, and only if they don't have, they are witnessed that there is no loss of function in the particular CYP2D6 enzyme, they are being prescribed with this medicine. So we have very few examples of how this genetic alteration or a small change in the genetic function is going to impact on the patient's medical treatment. But now we have a good data with us. That data can be analyzed in a perfect way so that we can come up with more viable solution, not only metoprolol and clopidogrel, but different drugs which have been taken in the market. And in another 2 years' time, my dear academic community, we are very sure that of course, the doctors will subject the patient to genetic diversity testing, and only from the results on the genetic diversity data, doctors are going to prescribe the drug. So this domain is going to witness a huge change. Of course, it is going to revolutionize the way in which treatments are being given now. Okay, I will give you a glimpse of what happens during an SNP analysis. So the first important step we need to know is that there are different tools which are available for the SNP detection. I will give you a glimpse of the different tools which are available. The best tool which is available in almost all multi-speciality hospitals these days is next generation sequencing facility, which can be effectively utilized for identifying the genetic variation. So the advantage is that large section of the genome, quick accurate detection of SNP databases. So we have a good platform in Illumina. Pacific Biosciences is doing a wonderful research there. We have Oxford Nanopore working on the technology. The second area which we are concentrating is we have SNP genotype array, right? Very small, simple chip, right? But you have millions of DNA probes specifically able to bind to different SNP alleles. So it is also very interesting data. Nowadays, currently it is being prescribed for all the non-communicable diseases, especially of high importance like cancer and diabetes, blood pressure, cardiovascular problems and all. But very shortly, last week we had an industry meet, and we had our collaborator discussing that for cosmetology, they are targeting on SNP data sets. Which particular treatment regimen will suit for a patient. There is a company based on Hyderabad. We are currently working in the company for related to the genetic testing facilities to establish here. And their product, one of the product which they are concentrating, of course, they were also targeting on cardiovascular diseases. What now they are targeting is a cosmetology domain or dermatology domain for hair treatment or cosmetological treatment. Patients are subjected to genetic diversity testing, and they are being prescribed with definite medications for a better for getting a better rate. So patients means public is aware, public knows that they need the best treatment which is available in the country, and they are ready to spend for it, and companies have started to utilize that advantage. And see, that's the power of genetic testing. So not that any means we are not going to be one drug for all, right? Whatever is good for me, I will test it, and then I am going to take it, thanks to the advanced technologies which are available right now. Then we also have a real-time PCR which is available out there, that is also available for identification of genetic variants. MALDI-TOF mass spectroscopy is also used for identifying these SNP variations in the patient. Okay, right. So now I have given you a glimpse of tools which are being currently used over there for genetic diversity testing. And when this genetic diversity testing is done, then comes the role of the bioinformaticians or the data analytic people who are currently working on automating the option. When the data is ready, they are going to automate. They are going to identify the hot zones in your genetic array, right? Which area requires immediate attention, and what is the genetic variation which is available in the patient. And for that, we require a set of intense tools which are available. Now, I'll be giving you a glimpse of all the tools which are available for once your data is available. Of course, we have the data available for the entire Indian population. Right? Okay, so you are going to work with any particular disease. If you are going to interested in working with cancer, or you are interested in working with Alzheimer's or any disease, and you want to identify whether the drug is good or not, whether it is going to affect a large population or not. Okay, good. The data is available. Now you can start working on that data with all the tools which you are going to discuss now. Right? So far, what we used is the database SNP, right? DB SNP was the tool which was previously used by all the researchers who are doing their excellent work on precision medicine. So DBSNP is actually maintained by NCBI, National Center for Biotechnological Information. It holds the data of all the genetic variants, whatever gene you are targeting on, whatever protein you are targeting on, whatever disease you are targeting on. Just go type the name of the target protein or the gene if you are very sure with the gene, which gene is responsible for that. Just type the name of the gene, it automatically gives you all the SNP data sets which are available. So that is a good, interesting fact to know about this particular case. So that is a widely used genetic database which is available, which has the data of all the diverse diseases. Any disease you take, the data will be available. Next is International Genome Sample Resource, right? It is like how we have a genetic data sets which are currently available in India. Similarly, we have a global data which is actually maintained by a consortium. It's an international consortium. It is hosted by both NCBI and EMBI, right? Both the agencies have focusing on it, and they are maintaining this international genome sample resources for people who are currently working on precision medicine. You can also go in this particular data set and use that for your research purposes. Okay. And there is one more interesting database which we call it as Farm GKB, right? GKB, it is maintained by Stanford University, right? So it also gives us several information. Like this is a web server, very interesting web server. They are concentrating on pediatric pharmacogenomics, and they are trying to identify the relationship between SNPs, drugs, and different phenotypes which are available. Right? So any drug you put, it will give you the data about the SNPs and phenotypes. Any phenotype, if you are aware of, you will get the information about the drugs, which drug will suit best or not. Those data is available in this website. So if you have time and if you're interested in doing your research in this particular area, if you want to know more about this particular area, you can note down this website. You can go into that website, get more details on that particular area. Right. So I have an interest. I have got the data. Yes, of course, genome data, India data is also available. So what is next required, right? How should I proceed further? So I will go give you a glimpse of what can be done next, right? So the important instruction first, after the data retrieval, as I told you, from different data sets, starting from DBSNP or Genome India database or international database, the next important thing what we have to focus is prediction of the deleterious SNP. This databases will give you the glimpse of all the SNPs. It is not going to be synonymous or non-synonymous only. It is going to give you the data of almost all the SNPs which are available. We need to start filtering it side by side. We need to start it filtering side by side. We need to predict the deleterious SNPs there. We have to first identify the non-synonymous SNP. Then we need to identify the deleterious SNP, which SNPs has the potential to create a definite impact on either the drug interaction or pharmacogenomics or whatever it might be. So for that, we have three important analysis tools which are available. One is first tool which we can use is SNP functional impact prediction tools. Second is we have a conservation analysis tool what we can use, and the third one is structural impact prediction tools. So there are a set of tools. Of course, I am going to give you the list of all the tools which are currently available and what people are doing research right now, and you can make use of that in your research if required. So these three analyses has to be done to identify exact deleterious SNPs which are creating a huge problem in that particular disease management. And once you have come up with the deleterious SNPs, yes, of course, you can do a protein structure modeling. You can model the mutated protein, and that mutated protein can be subjected to molecular docking to check whether the drug protein interaction is going to be the same or whether the drug protein interaction is going to change or not. And only then you need to identify the potential inhibitor which can be prescribed for the patient and not before that. So any disease, in another 2 years' time, everybody are going to be subjected to this form of treatment. Only after the genetic testing is done, they will be prescribed with the medicine, and that data will be available. Right, I'll show you an example of a data which our intern has worked with, and of course we have hidden few results because it is under the verge of publication. So this is how the interface looks like. Right, so once you go into National Library of Medicine, you can go there, just click on the SNP database and type the gene whichever you are interested with. So when you are typing this result, you will get approximately, in my case, I got around 7,301 genes. Right, out of the 7,301 gene, very little, my merger number were related to non-synonymous. Of course, it gives you all the data, but what we are intended is to identify the non-synonymous data. We have filtered the non-synonymous data. So the non-synonymous data is actually around 7,301 for a single gene, right? For a single gene with respect to a particular melanoma cancer, right? So this is the data. imagine the data with multiple disease conditions. So whichever diseases you are also interested with, you can just go and type the gene or the protein target which you are interested with, you'll get the data. So for a single gene, there are 7,301 non-synonymous SNPs which have been reported, and that data needs to be addressed. So this is the percentage of the different SNPs which are available. Right, synonymous SNPs accounts to 9.20% of the total result for that particular gene. Right, followed by non-synonymous SNP, we have around 21% of the genes which have been reported were found to be non-synonymous SNPs. Then few SNPs are in 3' untranslated UTR segments, 5' UTR segments, and there are also some synonyms. 50.70 are found to be synonymous SNPs. So our genetic makeup is getting mutated day by day, but definitely most of the situation, almost 50 to 70%, it is not going to create an impact on the public or the patients, but definitely there is a 30% chance that it could create a change on the protein, and of course the drug is not going to be the same for all, right? So once you have come up with the percentage of the sequences which are found to be causing a severe impact, the next process is like we can just work on the tools which are available to identify whether we will have a deleterious effect due to the not synonymous SNP or not. So I have listed a definite set of tools. Of course, you can use all the tools in your paper or your research, what you're doing. Shift is a wonderful tool, right? It is based upon sequence homology and evolutionary confirmation. So you can just give the protein sequence, and this is the deadline, means this is the limits, which indicates whether that SNP can be tolerated or it is going to have a deleterious effect. So this shift will give you an idea whether that variant will affect the protein function or not. So all researchers who are going to do research into deleterious effect of a non-synonymous or any papers you refer, the first tool of preference will be shift. And polyphen is also there. Polyphen is also a similar tool which will predict the possible impact of the variants on protein structure and function. It gives you a code like whether this SNP is benign or damaging or probably damaging. So based upon that data, you can further classify that protein, whether the SNP data is damaging or not. Then we have Provian. It is again an evolutionary conservation based identification, and it gives you a score whether your SNP is deleterious or neutral, right? Then you have PhD SNP. It's a SVM based machine learning approach. It will predict whether it is a disease causing mutant or a neutral mutant. Then we have Panther available out there, which is all about phylogenetic trees, evolutionary conservations, evolutionary relationship, and all those stuff. And we'll give you a functional annotation of the protein and SNP prediction, what functional effect it is going to have. We have SNP and Go, right, working on evolutionary information, gene ontology, and all those segments. It will predict whether the disease-causing SNPs are going to cause any problem in gene ontology sections or not. So this is the output of what your polyphen data will look like. Once shift is over, we will go for a polyphen analysis, and polyphen analysis will give you a data like whether it is probably damaging or not. This is the influence of polyphen. This means the result of, I have just given you a small snapshot of the result, what I have got from polyphen. It will give you a score whether it is going to be damaging or not, right? So in my case, the score is close to 1. It means that it is probably damaging. So that particular mutation, that at the position 527, instead of amino acid R, it is going to be H, right? So if that is the case, right, then definitely it is going to have a severe impact on the patient impact. So such data can be retrieved easily from polyphen data set. Okay, once that is done, we go for an evolutionary conservation analysis. How far the sequence is conserved in an evolutionary way, whether it is really going to be damaging or not, or is it a fast evolving site or not. So for that analysis, we can use different sites, like conserve is used. It will identify the functionally significant region. PhyloP is used. It will measure the conservation at nucleotide level, very specific, based upon the p-values. You'll get a p-value, but that will indicate whether it is a fast evolving site or not. Then you have JERP, right? It will identify the constrained regions in your genome, and it helps you to identify the functionally important elements. We have 5ml. It is also constructing phylogenetic tree. So these are all the tools which are currently available, and we are also working with a web server for a particular disease. We are coming up with a proposal very shortly. It will be launched online, and means for people to easily identify disease specific what impact it is going to create with all the outputs which I have listed right now. Then we have FAT const, right? We have MAFT. Rate Foresight, Frank, several sites are available to identify the genetic diversity and evolutionary conservation data for all the data sets which are currently available. Right? This is how a result look like, right? You'll get an output like this when you're working in this table. So this color scale gives you a conserved domain region. This color scale will give you an average color region, and this will give you a variable color zone, right? So these are the zones. So once you submit your data set, especially the SNP affected sequence in this particular data, it will give you which sites, right? Which sites are less conserved, which sites are mixed conservation, which are conserved and exposed, right? So you can see here 601 to 611, it is in a dangerous situation, right? You don't have any conserved region out there. The color code itself tells you, the dark color gives you a code that is conserved. You cannot see any conserved region out here. So this the amino acid position 601 to 610 is definitely going to be danger. So this protein needs immediate attention, right? So those details need to be studied and evaluated and has to be compared with your result. Okay, once you have done a functional analysis, then we go into mutational impact prediction. For that, what our team does is that we have iMutant and MuPro. So these are the different tools which we use currently. One is SVM based and another one is neural network based. This gives you a score, probably a delta G value score. That score will give you an idea whether the stability is going to increase or the stability is going to decrease based upon the mutation data which is available or not. So if the stability of the protein is going to decrease automatically, right? We need to work on that protein. We need to identify the disease mutation. We need to model the protein, and we need to find some drugs for that particular disease. I'll share your data where I checked with mutation for a melanoma cancer. These are the different RSIDs, right? SNPs will be usually listed in form of RSIDs in NCBI. Out of the 7,601 synonymous non-synonymous SNP, what we have filtered, only 5 are found to be dangerous, right? Filtered. We out of the 7,000, we use different tools. Whatever I have listed there, we have used all those tools, and definitely only the people with these 5 genetic variants are going to have a very damaging effect. And of course, iMutant also gave the same result. MuPro also we got the same result. The prediction was the same, but the index was different. MuPro used a neural networking scheme and provides a score out there. iMutant provides you an reliability index data, right? So that data will give us an indication whether this RSID is suitable to work or not. So out of the 7,507 repeated reported SNPs, we have got around 5 SNPs which are found to be very dangerous, right? Once that is done, we went for another website. It's also a web server. Anybody interested can also work. So we want to study what changes will be happening in the protein, right? It will give you a 2D representation of how mutation is going to have an impact, right? So I have deposited my sequence, which is mutated, right? I have taken the structure for all the 5, but I have given you the result of only 1. Right? So this is a mutation of an aspartic acid into an asparagine at the position 18. See the structure, how it changes, right? Okay. Right? So your structure is going to have mutated into this. So if in a 2D structure, if you are getting such a vivid result, imagine what will be the case in the 3D format. Definitely the structure of the protein is going to change exceptionally in your 3D structure. When that is going to change, automatically what happens? The drug cannot go and bind to the protein. When it cannot go and bind to the protein, of course, it is going to have a severe impact. So this is a mutation in the same protein at the position 86, right? I have a tyrosine converted into a cysteine. Again, see the 2D structure change, right? Okay, 309 at the position converted into alanine at the position 93. A shift in the position, right? So these changes can be expected in the protein. This Project Hope server will give you an idea of how the 2D representation of the protein will look like when the particular protein is being mutated. Okay, when that is done, the next step for us is to model the mutated protein, right? We are going to model because we have the data available with us. We know which substitution is out there, right? So that data will be given as an input, right? And we can model the particular protein. We have modeled the protein for all the particular, we have checked the Ramachandran plot outliers and all those things. We have corrected it actually before our study, and we have taken it further for docking. So we were actually working on data like what you can call. We are actually working on herbal alternatives. So in that particular form of research, we are identifying a particular phytochemical composition. That was a work which is concentrating on netopharmacology approaches also. So we did the analysis on that particular tool, and we filtered the tools means drug-like candidates, and of course all the 5 important molecules were taken: camphorol, epigenin, genistein, isorampin, aranjin. They all passed in that Lipinski rule of 5 or druggability analysis and ADME analysis. We tried to use that protein for docking, and out of that, camphorol was found to be suited best, right? So camphorol showed a least variation in binding affinity. That was the interesting part of our study. Though the number of hydrogen bond interaction varied, the binding affinity remained to be the same across all the RSIDs which have been reported. So this is the type of research which is now required in means drug discovery platforms, right? Identify a drug which can be which will be suitable for all the participants, irrespective of the single nucleotide polymorphism which the patient is subjected to. Just to check the entire data, identify a best suitable alternate which can be prescribed. Right? Imagine in another 2 years of time, you can witness all the pharmaceutical companies labeling that it is suitable for all genetic diverse. That means genetic participants or pool of genetic variants in India. Definitely, it is going to come in a very short span of time, because all the researchers are now having their focus on this particular domain, and they are trying to identify most suitable drugs which are better suited to all the or which with low variation in their binding affinity in all possible dangerous SNP sets. So we have also come up with some a very interesting molecule. I'm not revealing the molecule right here. Out of the identified molecules, again, that was also an elaborate study. We were filtering around 2,000 to 3,000 molecules in that particular study, and we have come up with one single molecule which suits against all the RSIDs or non-synonymous SNPs which are reported in that particular form of melanoma cancer. And that was the interesting part of our study, what we have got. So this is the end of the day. So if you are going to utilize this particular data set for your publication, then please, since we are into drug discovery research, we have employed all the tools, starting from identifying the databases. We filtered the data sets. Only 50%, means 27% was found to be lethal out of the 7,000 reported genes. Then we took it to the functional annotation tools, where we got around hardly around 8 to 10% of the genes which are in risk. Then we subjected it to mutational analysis. We did the 2D analysis and 3D analysis of the binding pockets on on the RSIDs which are subjected to mutation. We modeled the protein, and we tried to identify a most suitable drug which can best suit to all the conditions, and we get a wonderful result. And that data is being now shown to you for so this form of protocols, this form of procedures can be also used by you in your research to come up with some novel new drugs against a particular form of cancers or any such non-communicable diseases, deadly non-communicable diseases, which can well suit for the public need. And of course, our research needs an immediate attention in this perspective, because a drug need to suit for all, right? Though personalized medicines are coming up, any research has to have it should not have a negative impact on any genetic variant population which is available in our country. We also have done an effect analysis in a protein level, because we all know that there are tools available to identify the protein interaction, right? So in protein interaction, we have different websites which are available, which will give us an indication of how this protein network is going to change. So we also want to analyze that, okay, if there is going to be a mutational impact on the protein, what interaction Sorry, 1 second, I'll share the screen. Am I audible?
Yes, sir.
Yes, right. Thank you for that. Sorry for that inconvenience, and thank you participants for, right. So we want to actually analyze what impact is actually it is going to create on the protein to protein interaction. Whether any mutations happening in the protein is going to have an impact at the protein level or not, whether it is going to behave in the same way at the downstream level proteins or not. We want to identify that, and for that we used protein-protein docking tools. And in this particular study, we used a tool called as ClassPro. Most of you would have, if you are into bioinformatic research, you know that. We just use this tool to analyze how the protein is going to have a variation, how this mutant is going to behave in the downstream signaling pathways, whether it needs immediate attention or not. And this is a site called as ClassPro. You can just upload your PDB structures in that particular site. It will give you an output, and that data can be used for your research. So it gives us a result how much energy means, right? How much energy we are getting. So this energy will give us a result whether this mutational impact is going to have significant influence on the downstream proteins or not. And we analyze that also. And in our case, it is having a definite impact on the downstream signaling proteins also. So this protein needs immediate attention and needed to be worked with suitable drug molecule to avoid that particular issues. That part can also be taken into consideration and can be studied if you are interested. Right. So that's it. So that was the part of our study. And of course, before I wind up, just a few content with our labs. So we have a definite Academia Connect division where we are having MOUs with different institutions across Tamil Nadu, and we are into efforts putting MOUs with MOU, and we are also interested in working with countries outside our state. We are into first tie-up with the WR University. Very shortly we'll be coming up with the MOU out there. And we also do research consultancy, collaborative research, and student and faculty training programs as a part of our Academia Connect division, and I'm heading that division. We have several signature programs coming up on the part of our laboratory, like Academia Connect, where we go to the laboratories, colleges. We train the students in different upcoming tools. We give them an idea about the tools and how they can be applied in their subject or the research, whatever they are interested on. We also have a program called Lab on Wheels, where we go to different institutions with our research setup. We go establish there, train the students, and come back. We have a BioCareer Talk program, which is conducted almost every month, where different people from different industrial, like our collaborators and our industry connect people, will train the students on that particular area of their area of expertise. And that's it. That's about a short glimpse of what our Academia Connect division is doing. Since most of you are from the Academia division, I just wanted to inform that also. And thanks to the entire team, the management of Sedu Institute of Technology, Professor and Head, Department of Biotechnology, Nageshwari ma'am, and the coordinators, Dr. Amal and Prabhu sir, for giving me a wonderful opportunity to connect with the people out there. And thanks to ACT ATAL FDP portal for this wonderful program. I hope the audience would have got some insights on SNP data sets, their relevance, and how that particular form can be used for in their research, or how it is going to change the way drug discovery was in the previous situations, and how it will be taken forward in the upcoming era. Thanks to the participants and the organizers for this opportunity. Thank you. Over to the organizers. Thank you.
Thank you, Professor. Yeah, now the session is open to the discussion. I hope all the audience would have to the session. It is open to discussion. If you have any question, you may unmute yourself and ask your question.
Thank you, ma'am. Thanks for the feedback.
Sir, spare some time. Participants are getting ready. I mean, one of the participants trying to get question, but she could not connect it. Just a minute. She'll be asking his question, direct questions.
Yeah, they can also post in the chat box if there is difficulty in connecting. I will try to address it.
Yeah, if you have any question, please post in the chat also.
Thank you, sir. Thanks for the feedback. The Shakila ma'am, see, we are actually having a separate bioinformatics division. And, in that particular domain, we are providing training to students in recent areas. Like now, we are concentrating on NLP. We have been previously working on AI, ML, machine learning, artificial intelligence, deep learning platforms for drug discovery. Now we are trying to introduce neural linguistic programming also. And of course, we are working on different programming approaches. We are also into R programming. We do clinical SAS, all the tools which are currently out there in the industry. And if any students are interested in learning that, they're most welcome to our laboratory. We have a residential program where the students can come over here and get trained. Our campus is a residential campus, especially the Academia Connect division. So if they're interested, they can come over here, stay here and get benefited out of the internship programs. Or if you are interested in conducting a program related to that particular domains, then we are also very happy to come out there. We can organize such programs at your institutes for the benefit of your students. Maybe which area you are targeting, we will try to identify that. Yeah, protein interaction, Dr. Priyanka, of course, I told you, ClassPro can be used. Then we have String, we can use it. Cytoscape can be used. We have different plugins available in Cytoscape. So those can be also taken. Thank you, Lakia ma'am. Thanks for the feedback. I think since audio is not available for all, I think I have addressed your criteria. Shakila ma'am, no eligibility criteria. The students are interested, they are always welcome. Any aspiring student who is interested to learn the new techniques and get acquainted to the industrial experience, they can do it. Autodoc, yeah, Autodoc we do it not for covalent docking studies. The docking is almost non-covalent interactions. We are using Autodoc only for drug protein interaction studies. So that's it. We use Autodoc. We have Malgrow software. We can do in Malgrow, and we also have a version of Schrodinger available out there. So any collaborations also we are open. Thank you, sir. Thank you.
Sir, hello, sir. Your session was very informative, sir. And I have a doubt regarding what are the basic testing the patients are subjected to before the administration of the drugs. For example, you have mentioned that clopidogrel and before administration they have to go for some testing like
Yes.
so right to identify the CYP219.
Exactly.
Yeah, what type of basic test they have to undergo, sir?
So far, currently, we all know that doctors will take use of all the diagnostic tools available and the biochemical markers for prescribing the drug. Now, as I told you, now the concepts are changing. As I told you, we are also going to enter into genetic testing market very shortly. And we are going in tie-up with a particular laboratory where they are come up with a mini DNA lab. It's just a small setup.
Oh, okay, sir.
Nothing big in that. Normally, your DNA isolation chambers and everything, you will have a PCR machine inbuilt inside, and you will have a detection platform which is cloud attached. So, if you are going to subject the patients to clopidogrel, markers are available. You have the PCR components available. You can just go for a basic testing and find out whether the patient is susceptible to it or not. a normal PCR-based study is sufficient for us to identify whether the patient is susceptible to a particular drug. Right now, we are trying to identify the suitability of the patient towards a particular treatment.
Okay, sir. Okay.
And that is on the way to the market. Of course, it is there, but it is not penetrated widely into for public. And very shortly, I think in another 6 months' time or by end of this year, it will be penetrating to a wider range in the market. As I told you in the midst of my presentation, rather than acceptance in the medical domain, our device is getting acceptance in cosmetology and dermatological domain.
Okay,
Very
sir.
shortly, we expect that the same change is going to happen in hospitality domain also. So the entire way of diagnostic sector is going to change. That's
Okay,
it.
sir. Okay, sir. Thank you.
Thank you. So, no more questions. I think Dr. Amal, can I?
Professor, 2 more questions are there in the chat.
Yeah,
Can
Sorry,
you mention like the multiple database providing overlapping of results for assembly prediction tools during research? How far we can shortlist and focus on specific softwares?
Question from Dr. Saran? Or?
Ilakya, to the before.
Okay, there is a question from Ilakya, ma'am. Okay, there are multiple databases providing overlapping results for SNP-based prediction tool. How far we can shortlist and focus on specific softwares? See, it depends upon our objective. It depends upon our objective, which form of objective we are actually focusing on. Are we trying to identify any potential markers, or are we going to identify any potential treatment strategy? So for which application we are going to use, what is the algorithm behind the software, what they have done? That is why I have included the algorithm also during my presentation. So which algorithm is more reliable? We have to do a small homework on that particular area before using that particular software, right? Very stringent algorithms have to be used. Don't use multiple tools for a single application. Just check which form of algorithm is going to suit your need, which can give you a right solution. Try to use that particular application so that you can avoid that reliability issue that can be overtaken. And Sitendra sir, I have answered your question. Malati ma'am, yeah, Malati ma'am, internship is open throughout the year at Academia Division. And if you are any interested, you can just take my number. You can pass it to your students, or you can contact and get the information from us. Saran, yeah. After use, is there any validation needed? We are facing rejection. Yeah, of course, validation is required. We have to map it up with, I have not included that data here since I told you that we are into publication of that data. You have to do a proper validation trial also. But as far as SNP is concerned, mutating a cell in system is easy, but mutating a model in a cellular system is very difficult. So, of course, that is a challenge in this particular domain. But other than that, if you're good in silico work, please validate it with appropriate in vitro or in vivo studies, or else that is going to be. Even our interns are also trying to communicate the publication papers, which they are doing it here. Maybe it is art programming. They are doing multi-omic studies. They are doing single cell RNA sequencing analysis. But problem is that they have good data with that, but articles are not ready to accept it. Journals are not ready to accept it. And they are expecting for a validation in wet lab studies. They have a short validation, just a simple cell line study will do. If you have any small substantiated evidence stating that your theoretical study is good, then automatically your research will be published. I think that is the reason why, of course, not only you, we are also facing that issue of rejection in most of the papers. Thanks to all the participants for giving me an interactive session. Thanks for the good interaction out there. I think.
Yeah, thank you, Professor. Thank you for your valuable input and patient answering the questions of our participants. I hope everyone would have got enriched with the knowledge what you have shared. I by now, by this time, I'm just winding up the session 1, and participants will be starting our session 2 by sharply by 8 o'clock. So we have 10 minutes time. The same link you can join. Feedback link is posted on the chat. Kindly fill up your feedback along with which will be considered as attendance also. So thank you, Professor Anuf. We are closing the session. Yeah.
Thank you, Doctor. Thanks for the
Thank you.
opportunity.
Yeah. Take care. Thank you. So participants, please join here sharply by 8. We'll start our second session by 8 o'clock. Thank you.